Hello and welcome to the Spine Nerve Podcast. My name is Dr. Brian Joves.
And my name is Dr. Nicolas Karvelas.
And today we have a very special guest. We have Dr. Michael Fishman joining us to kind of talk about all things neuromodulation, but specifically about something that's kind of really caught the attention of the pain medicine world. Dr. Fishman, welcome.
Thanks, Brian. Thanks, Nick. Great to be on the podcast with you guys today.
We'll kind of jump into things because we have a lot that we want to get to today. But Dr. Fishman, if you have the opportunity and ability to, I would love to kind of hear how somebody as young as yourself has kind of made the foray into, clinical trials and research as much as you have, and specifically maybe as it's led to you being such an integral part of what's likely to be one of the most groundbreaking studies in recent history for pain medicine.
You know, Brian, that's something that I kind of pinch myself every day, and I feel very fortunate because when I come to work, there are so many balls in motion. There's clinical research that's occurring related to things that like DTM, which we're going to discuss in a few moments, but also other projects that we have here in the practice, a variety of different technologies, new startup companies that are actually, giving us the opportunity to get involved in designing studies and designing devices, and also, proving the efficacy and ultimately the cost effectiveness and at the end of the day, the impact on our patients of the existing therapies that we have. How I got involved in this is really, I just got lucky. I've had really great mentors along the way from my partner, Phil Kim, who when I came on board said, hey, we want to do research, but I'd love for you to figure out how to make that happen. And since then we've done, over 25 clinical trials, and we've built together a really great team of both investigators and coordinators. And we did that mostly on a homegrown basis with people from within our practice who just took on more responsibility and ultimately more training. So it really was just a private practice that started having an interest in research and ultimately built this, fairly robust clinical research arm to our practice. Now, today, what I would say is that it represents the collaboration between our group and Ricardo Vallejo and his team kind of happened, very organically. I was sitting at a dinner a couple of years ago, and I had no business being at that dinner, but Phil Kim was invited and he just brought me along. And I sat down and I'm talking to this guy about pulsed RF, which I'm very excited about. And it turns out that I'm talking to this guy, Vallejo R, who little did I know, I've been reading a lot of his papers and I'm spinning them back to him. And he's thinking, oh my God, this guy is talking to me about all of my papers. And we're just having this back and forth discussion. And, ever since then, we've had the opportunity to collaborate. And both Dr. Kim and I have had an opportunity to be part of the feasibility study and also the RCT now in DTM that has brought what I think are some of the, the most rigorous clinical trial results in neuromodulation for low back pain to patients and physicians across the world. So I'm very excited about that and that collaboration. And ultimately that journey is just literally being at the right place at the right time and probably having read the right, 50 papers to have that discussion. But at the end of the day, putting two and two together and talking to a like-minded individual is ultimately how we move the space forward. And I just, in some ways, was fortunate to be in the right place at the right time for that.
It's such an amazing story. I remember the first time that I heard it. Just, it is that kind of classic saying about just being prepared when opportunity knocks, and being ready to step up.
Yeah, and Dr. Fishman, I do want to say thank you, not only for your time today, but for your contribution to the field. We really do mean that. It's awesome what you've been able to accomplish for us, and we appreciate that. We obviously, the main focus of what we want to talk about today is the results of the trial. But real quickly, before we jump into the results of the trial, we were hoping, since you are, such an expert in the field of neuromodulation, we were just hoping real quickly, because in prior podcasts, myself and Dr. Joves, we've talked about to our listeners the different, mechanisms of action of neuromodulation and spinal cord stimulation. And since we have you, here with us today, we just thought it'd be a great chance to hear from your perspective, since you have such strong clinical as well as research background, in terms of what you feel like the best proven and most prevailing mechanisms of action of spinal cord stimulation is, and then perhaps how that might differ when we think about, DTM's spinal cord stimulation or differential target multiplex spinal cord stimulation?
Yeah, that's a really challenging question because, I'll be honest with you, when I think back about the history of neuromodulation, and I appreciate you guys giving me such credit, I really don't deserve the credit that you're giving me. There are so many people's shoulders that we all stand on. And what I'll tell you is that over the course of the last, 50 years in neuromodulation, we've seen an incredible amount of effort and work go into optimizing the delivery of the same therapy. That is paresthesia-based stimulation that really built upon the gate control theory and essentially, ascribed to the adage, no paresthesia, no anesthesia. And we saw a dramatic shift in our ability to treat back pain start with data coming in the 10,000 hertz range for back pain with anatomic placement, and also with lead placement at T910 with kilohertz frequency stimulation and a whole host of other frequencies, actually including tonic stimulation at that level. And that's been reported since the 90s and early 2000s by Baralat and up through the Caporal studies. Now, I will be honest with you, when I'm not totally sure that I fully understand the mechanism of action behind these therapies and some of the other, what we would call contemporary neuromodulation patterns. I've seen things that make sense to me, like Falowski's work looking at what the neuromonitoring signal or neuromonitoring responses to different waveforms. And we saw that there were some that had obvious physiologic responses, like the de Ritter formulation of burst. We saw others that had kind of just a mirror matching, parallel spike response with other burst formulations. I'm not sure what that means because to me, it's not an electrical phenomenon that we're dealing with. And I would say that the paradigm shift that Ricardo Vallejo's work with DTM really takes is that he looked at this biologically. And his view was, what am I stimulating and what happens when I stimulate those cell types? And I think that's incredibly important because as a pain physician, we all know that there is something about neuropathic pain that we can see, we can feel, we understand those features. There's a phenotype associated with it. And I think that Ricardo really took that phenotypic approach both in the basic science work. That is, he took animal models that had the same genes. He hurt some of them. He didn't hurt some of the other ones. Some got stimulated, some didn't. And he really evaluated what the biology of those animals was pre and post injury and pre and post stimulation with 1200 hertz, with 50 hertz, and also with this host of frequency pulse with combinations called DTM or differential target multiplex that targets the neuroglial interaction. And in that animal model, they had almost twice the improvement in recovery to the non-pain state in terms of mechanical hypersensitivity. And they also saw a very strong correlation across all of the cells that they targeted, microglia, oligodendrocytes, astrocytes, and neurons, that their biology, their phenotype was actually changing using whole genome sequencing and RNA expression profiling. And they were seeing that difference matching behavior and phenotype in these animals. 55 animals were in this particular study exposed to DTM as compared to 1200 hertz or 50 hertz. So when I start to think about biologically what that means when we translate it into humans, that's the most exciting, real, honest to goodness advancement in the field, is that we're designing a therapy based on a real rationale that we've measured and hypothesized should be effective, measured that it was effective in animals, and now testing it in a rigorous RCT in humans.
Yeah, and I appreciate you tackling that question so well. Sorry, I know it was a question was, you could spend years answering, but thank you for doing it so well. And that segues us into obviously the main thing that we want to focus on here, which is, with the recent release of the 12-month data from the DTM spinal cord stimulation trial, from your perspective, what do you think were some of the highlights and the key results that came out of that study in terms of the clinical response of these patients?
Yeah, so I think the key results are that first, these were typical SCS patients that were randomized in this study. So these were patients in your practice that were undergoing or planning to undergo a commercial trial of SCS. Everything was gone through insurance and paid for by insurance. The patients were, 60 years old. They had 10 to 15 years of pain, 1 to 2 previous back surgeries, baseline back pain, 7 to 8, baseline leg pain, 6 to 7. This is a very typical patient for SCS in my practice. And those groups of patients responded really well to stimulation, both the conventional stimulation patients and the DTM patients. And the primary outcome, which was three-month responder rate for back pain, was met with superiority for DTM. So at 3 months, 80% of subjects that were treated with neurostimulation, treated with DTM, had greater than 50% back pain relief, as compared to 51% of subjects with conventional stimulation. And that 3 month primary endpoint was superior. But, 3 months is a very, I think, small amount of time, especially when you consider a battery that has a 9-year warranty and a therapy that has to treat patients for years to come. So I'm very pleased that when we followed patients out to 12 months, we had an 84% back pain responder rate with DTM. That is 84% of subjects who were treated with neurostimulation, that is, who had a trial out to 12 months, had 84% responder rate, greater than 50% reduction in back pain. That's really a fantastic response. It gives me a great deal of confidence in this therapy. But what adds to that confidence is the fact that 69% of subjects, so 7 out of 10 subjects treated with DTM, reported profound relief of their back pain. So 7 out of 10 patients had greater than 80% reduction in back pain. And if you compare that to conventional stimulation, 35% of subjects had a greater than 80% reduction. And if you look back, if we were talking about 80% reduction in back pain in a third of subjects with conventional 20 years ago, we would be writing home all day about that. That would have changed dramatically the landscape of SCS. But I think today, the fact that we have both of these options available in the same system, really, really profound results with patients coming from a 7 to 8 back pain and ending at a 1.74, and coming with a 6 to 7 leg pain and ending with a 1.45 at 12 months. That durability in a system with, I think we can all probably agree is, from an engineering standpoint, one of the strongest neuromodulation platforms. Really fantastic mesh of best-in-class therapy and best-in-class technology.
Yeah, I think the most striking thing about this study, obviously the top line numbers are unbelievable, right? It is the kind of response that we've all been dreaming about for our patients for a long time. But like you said, even the traditionally treated patients had absolutely remarkable response. And so when we're thinking about, just sticking with what we've been doing for patients, the fact that we've come so far compared to, like you said, 20 years ago, I would argue even some of the trials from the late 2000s, or, kind of everything pre-Senza, would be really happy to have any kind of results in that arena. And so it's pretty impressive what you guys did. Can you walk us through maybe the thought process about the real world ideal of it? Obviously, as a private practice physician, seeing a study that's done that replicates our patient population means a lot to me, and to see this kind of study come through, in the power that it had, and as well as with the data that it produced. But what was your guys' thought on coming up with that design?
Well, so I didn't come up with the study design. I was really an investigator in this study. What I think makes the most sense to me, though, is that the patients in my clinic were truly treated in silos. That is, the Medtronic team that I work with every day commercially programmed the conventional group. And the Stimgenics team that was not owned by Medtronic at the time, completely separate company, managed the programming in the DTM group. And both teams did their absolute best to achieve good outcomes for the patients. And I think that is certainly reflected in the results we have in both groups. I think that there is also a value to that, in that these patients really, in many ways, were doing well across the board. As an investigator, looking at the 20-something subjects that we had in our clinic as part of the overall N, it was hard for me to tell what was better or worse, because frankly, most of my patients were doing really well in this study, as you can see. So to see that as an investigator was really fantastic, but I still had no idea until the results were officially released and published what the results would be across all 12 sites. And to see this kind of response echoing our clinical response in our, our site was really profound to see. I think they were worried, actually, because the conventional patients were doing really well, that they were worried that we wouldn't have shown superiority. But frankly, it's amazing to see how well patients did in the in the study. And I think that part of that has to do with technological advancements. I mean, tonic stimulation is there, there's optimization steps that I think have taken place in the technology to deliver it. Of course, that's not what we were testing in this study. But at the end of the day, this comparative trial is only comparative in clinical research, because in the real world, you have access to both these therapies on the same device for your patients. And going back to something you said before, DTM is within the on-label parameters of the Intellis device. So to do this study as the way it was done really added to the real world, implications. That is, these patients were not, subjects who would not have access to neurostimulation if it wasn't for clinical research.
And one thing you brought up, Dr. Fishman, was the durability. There was really no drop-off in terms of the improvement of these patients at the 12-month outcome. That obviously, for those of us who do neurostimulation, that's something we worry about in terms of some level of habituation that can potentially occur with these therapies. Do you, and this may, obviously we want to see further and further results, both clinically as well as from a research perspective in terms of these patients, but just kind of thinking about the mechanism of action of this, of the DTM spinal cord stimulation, in your mind, do you kind of hypothesize that we may not see as much of an issue in terms of habituation, maybe relative to other forms of spinal cord stimulation, or just wondering if you had a strong opinion or thought in regards to that?
I probably have more questions than I have opinions on this particular subject. What I can say is this. I think that the mechanistic work that underlies the principles behind DTM, which shows biological neuromodulation in rats, can only help us think better about what we need to measure in humans to understand how phenotypes change. And people have been thinking about different types of ways to do that, including non-invasive evaluations of different physiologic parameters and urinary biomarkers of things like NMDA agonists and other types of corollaries, because you are not going to have the same type of data that was used to develop DTM preclinically, because we're not going to have any humans that volunteer or that we would even ask to give us a portion of their spinal cord to measure the response. Is it possible that moving forward, things like evoked compound action potentials and other direct measurements of cord activity will help us start to understand whether habituation is related to being in or out of a therapeutic window, related to specific pathologic responses from the cord, related to just, I'll use a music analogy, using noise instead of, kind of conducting an orchestra to try and change the way the nervous system behaves. I don't know any of those things. I don't know if the preclinical work gives us anything more than questions about that. So I'm open-minded, and I would like to see people prove this to me, because at the end of the day, I want to know more objectively and more holistically, how am I improving the way patients' pathology is? But that would suppose or presuppose that we would have some way of measuring it objectively, like we do in the animal model. And we just don't. It's very challenging. Success with neurostimulation is individually defined. I think each of us in each different patient encounter we have general principles that guide what that means, but they're tailored to that individual and to that individual's treatment.
And so as we're working on kind of helping people to understand this clinical trial and helping them to figure out what this means, for their patients or patients that they may be, if they're, kind of a resident or a fellow, patients that they're working up and trying to find the best options for. How would you kind of wrap this up in a way that people who may not live in the world of neuromodulation like we all do can kind of digest it easily and kind of take it back to their institutions or to their patients and be able to help them all to understand?
Yeah, that's really. I'll take that in a couple of different veins, and I'm going to tell you this story in 1 minute. Okay. This is the difference between hardware and software. 50 years in neuromodulation improving hardware. Much of the hardware advancements were made by Medtronic. They developed the first neurostimulator. And just if you look at the history of neurostimulation, almost every first in man innovation has come from them from an engineering standpoint. When you take that platform and recognize, for those of us who implant lots of devices, that there's a lot of advantages to it, none of those advantages actually matter until you've laid on top of that a therapy that actually works, that works really well, and that you can use to target important targets like the low back, which is notoriously difficult to treat. DTM stimulation looks at pain from a biological standpoint and looks at spinal cord stimulation as what are we actually stimulating? All of the components of the spinal cord, which involve multiple different cell types. And so the hypothesis behind DTM said, okay, if we stimulate and target each one of these cell types in animals, can we have a better behavioral response? The answer is yes, we can. And does that behavioral response match the phenotype of those animals when we measure that objectively using genome sequencing and RNA expression profiles? Yes. And this RCT has shown that when we take patients in a rigorous, scientific, randomized controlled trial, and we apply two completely different therapies with two different teams to typical SCS patients, the ones who would be having SCS in my clinic no matter what, and those who are having primarily back pain, that we have superior outcomes, and that those outcomes are durable at 12 months. And in this comparative study, we've reported the highest responder rate for back pain in similar randomized controlled trials. That is RCTs with more than 100 patients randomized, comparing two different SCS therapies with 12-month outcomes. And is the difference between 81% and 84% material to you in your practice? No, it's not. What I will tell you is that this reflects my real-world results in my practice with the Intellis stimulator in both of these groups. And for back pain, 70% responder rate for 80% back pain relief sets a new bar in my practice for profound response. And seeing that in 7 out of 10 subjects is really fantastic. And knowing that I have conventional stimulation, because we place all of these stimulators with paresthesia mapping, because it's on label for Intellis to do so. But this just completely confirms to me the need to do that, because 35% of patients in the conventional stimulation arm had greater than 80% back pain relief. As a backup plan, that's amazing. So, you know, to me, this really is a huge advantage to my patients to be able to walk in the room and say, you know, these are the results of my practice because we were part of this study. And I would say to my colleagues, you should really see what this looks like in your practice, because at the end of the day, home run after home run after home run stops becoming novel and starts becoming the majority of your cases when your real world evidence matches your RCT. And I can share with you that in my practice, it does.
Which is, you know, I think it's an amazing thing for you to be so confident in and be able to say that. You know, I think just having been around this industry long enough and having had the opportunity to talk to a lot of people, it's not always been the case that people can display their real world data matching RCT data. And so I think that's, from my perspective, that's definitely something that I think is extremely exciting. You know, having heard that not only from yourself but also from the other investigators that have been doing it, you know, and starting to implement this within our practice as well. And so as we're kind of coming closer towards the end of wrapping up and making sure that we can make this easily palatable and digestible for everybody, are there, I know we've kind of covered the big broad strokes and a lot of the most important topics, but is there anything that we have failed to cover so far that you want to make sure that our listeners are able to take away and maybe even more importantly, know where to reach out and to get more information about everything?
Yeah, honestly, I think that the coolest thing about this is the fact that it works really well for patients, but that the story behind it is one that could be almost any of us. And when I say that, I mean this. You know, Ricardo Vallejo, he's a pain doc. He's treating these patients. He's trying to figure out what he's doing, and he's asking himself questions. And as an immunologist, he had the tools to get to this kind of point where he could ask those questions in a meaningful way and actually answer them. But I think we're getting pretty close to the point now where almost like everybody has an app on the cell phone where people can really start to have a lot more freedom in designing programming and designing experiments to understand the next programming breakthroughs in neurostimulation. The bar has dropped dramatically. You know, you don't have to develop a device anymore, right? Just like you wouldn't find any app developer out there going out and building their own cell phone or tablet. You know, the future of decoupling hardware and software is very rich for patients and physicians, because I think it really will open up the amount of innovation that we can, you know, display in this space. And I think that is huge. And if this shows anything, it's a template for others to take up that same charge and ask really important questions about what we were doing, why we were doing it, and then just test it. The barriers to testing these things now, it's just programming for the most part. It's extremely safe. And we have so much that we have to learn about neuroscience and the nervous system and pain. It's, you know, kind of a blue ocean, if you will, of opportunity for us to help patients and learn more about this very difficult and complex biopsychosocial disease.
That's a great way to wrap. Dr. Karvelas, any closing questions or thoughts for Dr. Fishman?
No, just thank you, Dr. Fishman, so much for your time. We appreciate it. And like I said, for your contributions as well.
Yep. Thank you very much.
Guys.
We appreciate it. And everybody, you can reach out to Dr. Fishman on all social media. He is a big fan and believer of hearing from his fans.
All right.
All right, guys. Stay tuned for those legal disclaimers.